Welcome back. Now in this section, I'll be talking to you about plasma proteins. What are plasma proteins? Proteins that are found in the plasma are plasma proteins. So where is plasma? Blood. What is the difference between plasma and serum? Serum does not contain fibrinogen. Plasma contains fibrinogen. How are they formed? How can we separate plasma and serum? If we add an anticoagulant to a tube while removing the blood, then after centrifugation, we get plasma. The clear fluid that is obtained is plasma. Now, when you have added a coagulant, what has happened? The blood has not clotted. So, we get plasma. Now, if we withdraw blood and put it in a tube without anticoagulant and then we allow it to clot and then centrifuge, we get serum. Now, most of the times, what we use for different procedures are serum and not plasma. But now I'll be focusing, most of the proteins that are present are anyway present in plasma. We'll talk about all of them. Now, the plasma contains nearly 100 different types of proteins. So, what is the total concentration of the protein? Now, these are values that you are supposed to remember. What we call as total protein concentration. Now, total protein concentration is what, one thing to be remembered here is proteins are in grams. Many of the things are in milligrams per dl, proteins are in grams per dl. So, the total protein concentration is 6 to 8 gram percentage of grams per deciliter or grams per 100 ml. Whatever be that, all mean the same. So, gram percentage or gram per 100 ml or gram per deciliter. All three mean the same. So, from now onwards, I will be using the term gram percentage. I won't be using gram per 100 ml or gram per deciliter. All three mean the same. Now, the total protein concentration, as I said, more than 100 proteins are there and the total protein concentration is 6 to 8 grams per dl. Now, the different plasma proteins, when we are subject the total proteins and we subject it to different techniques by which we can separate out the different proteins, and then we have got three major classes that are produced, that is albumin, and its concentration is, you must be knowing by now, 3.5 to 5 gram percentage. Again, I keep on repeating, 3.5 to 5 gram percentage. Now, the second important fraction that we get, which is clinically important and in the lab also it is important, and that is globulins. Albumin, globulin. Albumin and globulin are constantly being measured in the lab everywhere. So, what is the concentration of globulin? Globulins are 2.5 to 3.5 grams per cent. Now, values may vary depending on sometimes the method that is used for the estimation of the different proteins. So, each lab makes its own reference values, but this is the commonest thing that is usually seen all in different labs. Now, fibrinogen is another thing that will be present in plasma, and fibrinogen has much lower concentration. Fibrinogen is the only protein that is present in milligram percentage. So, we say that fibrinogen is between 200 to 400 milligram percent. Now, please remember, fibrinogen is not estimated so much in different laboratories because it is a serum which is subjected to the estimation, and in the serum fibrinogen is not there, hence we estimate albumin and globulin to know about the different disease conditions. So, to go ahead, one more thing, important thing to be remembered here is all the proteins are synthesized in the liver. When I say such a big rule like this, all are synthesized, every rule has an exception. So, which is the exception for this? Except the gamma globulins or the immunoglobulins. I'll be talking to you more about this later. So, all plasma proteins except the immunoglobulins are synthesized in the liver. Now, where are the immunoglobulins synthesized? We'll talk about it as we go ahead, mainly in the lymphoid tissues. Now, which are the different ways by which we can separate out the different fractions of the proteins, plasma proteins. We call these are called as salting out, electrophoresis, ultracentrifugation, immunoelectrophoresis. Various methods are there by which we can estimate different fractions of proteins that are present. For example, we have the BCG method, what we call bromocresol green method, for the estimation of albumin. We estimate total protein by different methods like Lowry's method, etc. And then all we do is subtract albumin from the total protein concentration, we get the globulin concentration. Now anyway, other than the different methods, one important method which is significant, which is required even from the exam point of view, is electrophoresis. Now it is the electrophoresis that I will be talking about. So when we subject serum to electrophoresis, I have shown you serum to electrophoresis. Please remember, even though we say plasma protein electrophoresis, I have told you the difference. So when we subject serum to electrophoresis, this is how the electrophoretic slide will look like. Now if you look at it, we always remember how is electrophoresis performed. When we subject it to an electrical field, charged particles will move either to the anode or to the cathode depending on the charge present on these proteins. So based on this simple principle, we can have an electrophoretic method for separating the serum proteins. So when we put a small strip of filter paper dipped in serum, when it is applied and it is put on an agarose gel or any other support media and electric current is passed through it, we find that the plasma proteins or the serum proteins separate out. Now they separate and form different bands. Now what is important here is where they migrate to, what comes one after the other, the concentration. Now albumin is the one which migrates fastest and it goes most to the anode, and that is the band which is closest to the anode is albumin. After albumin we have all the globulins. The globulins are alpha 1 globulin, alpha 2 globulin, beta globulins and the gamma globulins. We'll talk about each of these fractions one by one. Now, as I said, the major classes of plasma proteins, I first told you it is albumin which goes towards the anode. Now, in certain cases, what happens is, if the separation is very good, if the technician is good, if the technique applied, everything that is required for electrophoresis is good, sometimes we get a band which moves faster than albumin, further than albumin, and we call this band as the pre-albumin. So it is a minor band. It is usually seen only if the separation is good. Now, which are the proteins that are present in this pre-albumin band? We include transthyretin and thyroxine. Transthyretin and thyroxine, retinol binding pre-albumin. Transthyretin is the one which is responsible for the transport of the hormone thyroxine. Retinol binding pre-albumin is the protein that is responsible for the transport of vitamin A. Now this is only if the separation is good. If the separation is not good, both these proteins will be a part of other proteins only. It won't be separated out. Transthyretin which transports thyroxine and retinol binding pre-albumin. So this is important from an exam MCQ point of view. Now if you go further, the next class that we will be seeing is albumin. Now where is it synthesized? I have already told to you, it is synthesized in the liver. Its normal value is 3.5 to 5 grams per person. Its migration, it moves fastest towards the anode. Now different functions of albumin, you all will be knowing about it. One of the most important function is it is responsible for maintaining the colloid osmotic pressure. It is responsible for the transport of fatty acids, bilirubin, calcium, steroid hormones, drugs. All these are transported by albumin. It has a good buffering capacity. Always remember, whenever I say a protein has got a buffering capacity, I mean to say it is mainly the histidine residues which are present in the protein which are responsible for the buffering action of that protein. I keep repeating that. Now, other than that, the other function of albumin, it is responsible for the transport of essential amino acids which are required from the liver to the extrahepatic tissues, to wherever they are required. Whichever amino acid is required, albumin will help in the transport of these essential amino acids. Now we go on to the clinical applications regarding this band. Now, hypoalbuminemia is the condition when the albumin levels are decreased. So, whenever the albumin levels are lower than 3.5, do we need electrophoresis to see whether it is the albumin levels are low? No, we do not require. We have other methods by which we can estimate albumin, and then we can tell whether the albumin levels are low, or usually it is the low conditions when it is important, and hypoalbuminemia is what we call it. Now, I'll be talking about the different causes and effects later on. Now, one important thing about this is the drug interactions that can take place depending on the albumin levels. Now, phenytoin and dicumarol are two important drugs which can interact. Now, their interaction depends upon the albumin level. Now, albumin also has important role in calcium metabolism. What I could like to say is, whenever the total calcium levels are, if you get a total calcium levels are low, it might be because of various different reasons. Now, if the total albumin levels are low, the total calcium levels will come low, but the patient will not be having low calcium levels. Why? Is free calcium is the one which is responsible for the actions of calcium, and this free calcium, the ionized calcium, is levels will be normal, and hence there will not be any tetany in case of if the total calcium low is because of hypoalbuminemia. Now, hypoalbuminemia, there are lots of causes. I have listed down a few of them. Some of them you might be have learnt it in different conditions also. Let us look at it. Now, cirrhosis of liver. Why cirrhosis of liver or any other liver condition where liver is malfunctioning? What happens is albumin is synthesized in the liver. So, it is not being synthesized. It leads to hypoalbuminemia. Nephrotic syndrome. Why in nephrotic syndrome? Again, in nephrotic syndrome, the albumin is excreted out of the body in the urine. Decreased intake like protein energy malnutrition, or it may be because of decreased absorption of albumin like in protein losing enteropathy. Whenever albumin levels are low, the first thing that we see is edema. Then it is responsible for different drug interaction. As I said, phenytoin and dicumarol interactions to take place when if the albumin levels are low, and calcium is one more important thing that is required to be remembered when we talk of albumin. So we go on to the next band. Which is the next band? The alpha-1 globulins. Now, in the entrance exam, most of the time, the following are the proteins which belong to the alpha-1 globulin, except or which of these proteins belong to the alpha-1 globulin. Now, it is very difficult to remember which protein goes into which band. So, there are certain ways which you can remember. Remember, alpha-1 globulin, the minute the word alpha-1 comes, you need to have something which is alpha-1. I have told to you already one very important enzyme and a very important protein, and that is alpha-1 antitrypsin. Alpha-1 antitrypsin. The other alpha-1 which you can think of is alpha-1 fetoprotein. Alpha-1 fetoprotein. So these names itself will tell you that these prefixes itself will tell you that they belong to the alpha-1 globulin. So other than that, which are the other proteins which are difficult for you to remember? They include the retinol binding protein. What does this retinol binding protein do? It is responsible for the transport of vitamin A. The other is prothrombin. So these are the, I think, most important proteins that are present in the in the alpha 1 fraction that you will have to remember. There is also HDL, high density lipoprotein, which is also an alpha 1 globulin. So these are the main important proteins present in the alpha 1 globulin fraction. Now I have got given here the clinical importance. AFP, alpha 1 fetoprotein, can be measured in the amniotic fluid for the diagnosis of open neural tube defects and Down syndrome. So it is used in the prenatal diagnosis of Down syndrome or neural tube defects. That is when in the amniotic fluid AFP levels will increase. AFP is also used even in adults as a tumor marker for hepatoma. Normally, hepatic cells will not secrete alpha fetoprotein, or even if secreted, the levels are very low. But in case of a tumor, a hepatoma, the alpha fetoprotein levels are increased. I have already told to you about alpha 1 antitrypsin, which genetic deficiency leads to emphysema. Prothrombin time is used as a liver function test. So we go on to the next category, that is alpha 2 globulins. Again, as I said, this is important only from alpha 2. This is important from an exam point of view. Minute I say alpha 2, you need which are the proteins? One is the alpha 2 macroglobulin, because the prefix is there. So which are the other proteins? How to remember them? So there is some mnemonic, what we call CHTT. So which is these proteins? Includes ceruloplasmin. H stands for haptoglobulin. I think I'll show it on the slide itself. Ceruloplasmin. H stands for haptoglobulin. T stands for transcontin, and one more is for thyroxine binding globulin. So ceruloplasmin, transcontin, haptoglobulin and thyroxine binding globulin. These are the main 5 proteins that are present in the alpha 2 fraction. Again, this is important just as a recall based fact in a MCQ. So these are the proteins present in alpha 2. Coming on to their clinical significance, what is the main function of ceruloplasmin? Ceruloplasmin is important in Wilson's disease. Why? In Wilson's disease is a disease in which copper metabolism is affected. I'll talk about it in copper metabolism right now. So please remember that nearly 90% of the body's copper is present along with ceruloplasmin. Some 10% is present along with albumin. What is alpha 2 macroglobulin? Alpha 2 macroglobulin is a in vivo anticoagulant. What is its importance? Alpha 2 macroglobulin is increased in nephrotic syndrome. Now, other than that, as I said, haptoglobulin. What is the main function? Hapto means what? Hapto means it binds to hemoglobin. That's why it prevents the loss of hemoglobin from the body and thereby prevents the loss of iron. So body wants to conserve iron. The body does not want to lose iron at all. So in case there is a hemolysis, or even if there is a senescent RBC, this haptoglobulin will go and bind to the hemoglobin and prevent its loss. And it is also an indicator. Haptoglobulin levels are an indicator for whether there is any hemolysis that is taking place within the body. Alpha 2 macroglobulin, as I said, is increased in nephrotic syndrome. So this is the alpha 2 globulins. We go on to the next class, and that is the beta globulins. Again, as I say, it is just a matter of recall. That is why you need to know this, which of the different proteins are present in which different fraction. So the beta globulins. Now, in certain books, you may get beta 1 or beta 2, but the whole fraction separation is very difficult usually. So that's why at this level, we combine all of them and put them as beta globulins. Again, we include the CHT even in this. So there is always a confusion, which is the C, which is the H, and which is the T. Now, CHT again stands for, if you can remember, please do remember. C stands for C-reactive protein, CRP, what we call as C-reactive protein. H stands for, there in alpha 2 globulin you had haptoglobulin, here we have hemopexin. Again, this is related to Hemopexin is related to hemoglobin, something related to heme itself, but hemoglobin itself, here this prevents a loss of heme from the body and thereby prevents a loss of iron from the body. And it also includes, since we are talking of iron, it includes transferrin. Transferrin is the protein which is responsible for the transport of iron. Now other than that, we have LDL and beta 2 microglobulin. I have just got importance of this. Hemopexin binds to free heme. Transferrin transports 2 molecules of iron, and C-reactive protein is very useful in differentiating bacterial from viral infection. Other than this, C-reactive protein is nowadays can be estimated by a high sensitive reaction which we call as HSCRP. This is useful in knowing about coronary artery diseases. I'll be talking about it later when I talk about the disease. So HSCRP is very important. Now other than that, now we finished alpha 1, alpha 2, beta. The other important globulin is the gamma globulins. Gamma globulins do not migrate, they will be near the origin itself. The gamma globulins include the antibodies. They are produced, all other proteins are produced by the liver except gamma globulins, which are produced by the lymphocytes and the plasma cells. Structure of immunoglobulin, most of the immunoglobulins are glycoproteins, meaning they are proteins which are some glycation, carbohydrate chain. They have two types, light and heavy. Light chains have lesser molecule than the heavy chains. It is a Y-shaped molecule. Basically, it is a Y-shaped molecule made up of 4 polypeptide chain, 2 H chains and 2 L chains, which are all linked by disulfide bonds. Now, depending upon the heavy chains, we have other types like IgG, IgA, IgM, IgE, IgD. We will be learning more about this in microbiology and in immunology. This is just to show that domains are formed, in different domains are present in each of these immunoglobulin chains. Now another protein is fibrinogen. Fibrinogen, as I told you, is present in plasma and not in serum. It is an acute phase protein, and fibrinogen is converted to fibrin during blood clotting. Fibrinolysis is, in vivo fibrinolysis is constantly being done by plasmin, by the protein plasmin. Now I use the term acute phase protein. So what does it mean? It means a response, a non-specific response following any infection, trauma, inflammation, burns, anything. It is an acute phase response. So whenever such a thing happens, the body responds either by decreasing the concentration of many proteins, we call it as the negative acute phase reactants, or increasing the concentration of certain proteins, we call it the positive acute phase reactants or the acute phase proteins. All this, albumin, pre-albumin, transferrin, are negative acute phase proteins. Is this important? Yes. From a exam point of view, which of these are negative acute phase proteins? And CRP, C-reactive protein, ceruloplasmin, alpha-1 antitrypsin, fibrinogen, haptoglobulin are the positive acute phase proteins, are the acute phase proteins.